Hello friend and welcome to Facts for Sleep. Tonight we are going to explore one of the most remarkable processes in the natural world. It is a process that happens slowly, quietly, and almost invisibly. It happens in burrows beneath frozen ground, in hollow trees, in caves tucked into mountainsides, and in the mud at the bottom of still ponds. It is a process that turns living creatures into something that barely resembles life at all. We're going to spend this time together exploring the quiet science of animal hibernation. If you find this kind of slow exploration helpful, you may want to subscribe. New videos arrive regularly, and each one is designed to accompany you gently into rest. To understand hibernation, it helps to begin with a simple idea. Every living animal needs energy. Energy comes from food. Food is not always available. In the warm months of the year, food is often plentiful. Insects fill the air. Plants produce fruit and seeds. Fish move through rivers. The world is generous. But as the days grow shorter and temperatures fall, that generosity begins to fade. Insects disappear. Plants stop producing. The ground hardens. Rivers slow beneath a layer of ice. For many animals, finding enough food to survive the winter would require more energy than the food itself could provide. Over millions of years, a different solution emerged. Rather than searching for food that is not there, some animals learn to stop needing it entirely. They learn to wait. And the way they wait is through hibernation. Hibernation is not simply sleep. This is one of the most common misunderstandings about the process. When a human being sleeps, the body remains warm, the heart continues beating at a normal rate, the brain stays active, and the person can be woken easily. Hibernation is something far more extreme. During true hibernation, the body undergoes a controlled and dramatic reduction in almost every biological function. The body temperature drops sharply. The heart rate slows to a fraction of its normal pace. Breathing becomes so slow and shallow that it is barely detectable. The animal enters a state of suspended animation that can last for weeks or months. It is not sleep. It is closer to a kind of biological pause. The word hibernation comes from the Latin word hibernus, which means wintry. The Romans observed animals disappearing in autumn and reappearing in spring, and they understood that something unusual was happening, even if they could not explain the mechanism. For centuries, people believed that swallows hibernated at the bottom of ponds. This idea was popular in Europe well into the 18th century. Gilbert White, an English naturalist writing in the 1700s, debated seriously whether swallows dove into the mud of ponds each autumn and re-emerged in spring. We now know that swallows migrate rather than hibernate, but the observation that prompted the theory was real. Animals do disappear in winter, and they do return. The question of where they went and what happened to them took a long time to answer properly. The scientific study of hibernation began in earnest in the 19th century, when naturalists started making careful observations of animals in captivity and in the wild. They recorded body temperatures, they counted heartbeats, they measured the breathing of animals that had entered their winter rest. Slowly, a picture emerged of a process that was far more complex and far more precisely regulated than anyone had imagined. Hibernation was not a passive collapse into unconsciousness. It was an active, carefully orchestrated physiological transformation. One of the first things scientists measured was body temperature. In a normally active ground squirrel, the body temperature sits at around 37 degrees Celsius. When that same animal enters deep hibernation, its body temperature can fall to just above the freezing point of water. In some species, it falls even lower, to temperatures that would kill most mammals outright. The animal's body does not simply lose heat by accident, it actively lowers its own temperature as part of a controlled program. Special proteins and biochemical signals guide the process step by step. The body becomes, in effect, its own thermostat set to the lowest possible setting. Because of this dramatic drop in temperature, the chemical reactions inside the body slow enormously. This is a basic principle of chemistry. Cold temperatures slow chemical reactions, and warm temperatures speed them up. The body of a hibernating animal is running its chemistry at the slowest possible rate while still remaining alive. Enzymes work slowly. Cells consume very little oxygen. Waste product accumulate at negligible rate. The animal has become, in a very precise sense, almost still. The heart reflects the stillness. In a hibernating ground squirrel, the heart rate falls from around 350 beats per minute to as few as 3 or 4 beats per minute. In a hibernating black bear, it falls from around 55 beats per minute to as few as 8. In some of the most extreme hibernators, like the Arctic ground squirrel, the heart may beat only once every few minutes during the deepest phases of torpor. Blood moves through the body so slowly that it pools in certain areas, and circulation to the limbs is drastically reduced. The animal's extremities become cold and stiff, yet the vital organs continue to receive just enough blood to remain functional. Breathing slows in parallel with the heartbeat. A hibernating woodchuck, which is also called a groundhog, may breathe only once every 5 or 6 minutes during the depths of winter. In the warmer months, the same animal breathes around 16 times per minute. This reduction in breathing is not dangerous during hibernation because the body's need for oxygen has fallen so dramatically. The cells are doing so little work that they require almost no oxygen to sustain themselves. The lungs, the heart and the blood work together at a minimum level, each supporting the others at the lowest sustainable pace. The brain also changes during hibernation. In an active mammal, the brain consumes an enormous proportion of the body's energy. It generates constant electrical activity, processes sensory information, regulates body functions, and manages movement. During hibernation, brain activity falls to a very low level. The electrical signals that pass between neurons slow and diminish. The animal cannot be easily roused by sounds or smells. In very deep hibernation, the animal is almost entirely unresponsive to its environment. Yet the brain is not dead. It is, like everything else in the hibernating body, waiting. The energy that sustains all of this slow activity comes from fat. Before entering hibernation, animals spent weeks eating intensively. This period of intense eating is called hyperphagia, from Greek words meaning excessive eating. A black bear in hyperphagia may consume 20,000 calories in a single day. A ground squirrel may double its body weight over the course of a few weeks. This fat is stored primarily beneath the skin and around the internal organs. Once the animal enters the hibernation, this stored fat becomes its only fuel. Over the arse of months, the fat is slowly metabolized, releasing energy at a rate just sufficient to keep the body alive. The metabolic rate of a hibernating animal can be as low as 1% to 5% of its normal active rate. This extraordinary efficiency is what makes hibernation possible. An animal that normally needs hundreds of calories a day may survive an entire winter on the energy stored in a few kilograms of fat. The body has become so frugal in its energy use that it can sustain itself for months on what would, in normal activity, last only a few days. There are different types of animals that hibernate, and not all of them hibernate in the same way. Scientists make a distinction between obligate hibernators and facultative hibernators. Obligate hibernators enter hibernation on a reliable schedule, regardless of environmental conditions. Their hibernation is driven by internal biological clocks and hormonal changes that follow the seasons. Even if an obligate hibernator is kept in a warm laboratory with abundant food, it will still show a tendency to reduce activity and lower its metabolism in autumn. The drive to hibernate is built deeply into its physiology. Facultative hibernators, by contrast, enter hibernation primarily in response to environmental cues like cold temperatures and food scarcity. They hibernate when conditions make it necessary, but not necessarily on a fixed schedule. Bears are often cited as examples of animals that exhibit a form of facultative hibernation, though as we will explore, bears present a special case that sits somewhat outside the usual definition. Among the most studied obligate hibernators are the ground squirrels. There are many species of ground squirrels distributed across North America, Europe, and Asia, and several of them are among the most extreme hibernators known to science. The Arctic ground squirrel, which lives in the tundra of Alaska and northern Canada, enters hibernation in late summer or early autumn and remains in hibernation for as long as 8 or 9 months. During the deepest phases of its hibernation, its body temperature falls below 0 degrees Celsius. Its tissues do not freeze because they contain substances that lower the freezing point of water, but the animal is, in a real sense, colder than the frozen ground around it. It is among the only warm-blooded animals known to allow its body temperature to fall below the freezing point of water and survive. The Arctic ground squirrel does not remain in this frozen state continuously throughout the winter. Every 2 to 3 weeks, it undergoes a remarkable event called an arousal. During an arousal, the animal's body temperature rises rapidly back to near normal levels. Its heart rate climbs. Its brain becomes active. The entire process of warming takes several hours and consumes a significant portion of the animal's stored fat. The animal shivers intensely as its muscles generate heat to warm the body. After spending between 12 and 24 hours at normal temperature, it cools back down and returns to deep torpor. This cycle of torpor and arousal repeats throughout the winter. Why do hibernating animals arouse at all? This question has puzzled researchers for decades. The arousals are costly in terms of energy. Each arousal consumes more fat than the animal burns during an equivalent period of deep torpor. If the purpose of hibernation is to save energy, then waking up periodically seems counterproductive. Several explanations have been proposed. One is that the brain and nervous system accumulate changes during deep torpor that must be corrected during arousal. Deep torpor appears to impair certain aspects of cellular function, and the warming period may allow the body to repair those impairments. Another explanation involves the immune system. During deep torpor, immune function is severely reduced. Periodic arousals may allow the immune system to become briefly active and address any infections or cellular damage that have accumulated. A third hypothesis relates to sleep itself. This is one of the more surprising findings in hibernation research. During deep torpor, despite appearances, the animal is not truly asleep. Its brain waves during torpor resemble wakefulness more than they resemble sleep. When the animal arouses from torpor, it almost immediately enters what appears to be genuine sleep, complete with the slow-wave brain activity associated with deep sleep in normally active animals. Some researchers have proposed that hibernating animals arouse periodically, partly because they need to sleep, The torpor state, despite looking like sleep to an outside observer, does not possess the restorative brain processes that sleep provides. The animal wakes up in order to sleep properly and then returns to torpor. This idea has intriguing implications. It suggests that sleep and torpor, despite their superficial similarities, are fundamentally different states. Sleep is an active process that the brain requires for maintenance. Torpor is a different kind of state, one that conserves energy but does not substitute for sleep. The two must occur separately. An animal cannot hibernate its way through the need for sleep. The preparation for hibernation begins long before the first frost. In late summer, as day length shortens, the bodies of hibernating animals begin to change. The shortening of days, detected through the eyes and processed by a region of the brain called the suprachiasmatic nucleus, triggers hormonal changes that set the preparation in motion. Melatonin, a hormone produced in the brain's pineal gland in response to darkness, plays a key role in signaling the approach of a winter. Rising melatonin levels in autumn help trigger hyperphagia and the physiological changes that prepare the body for torpor. Alongside the behavioral changes, the body's biochemistry shifts in preparation for the extreme conditions ahead. The composition of cell membranes changes. Cell membranes are made partly of fatty acids, and the ratio of saturated to unsaturated fatty acids affects how fluid the membrane remains at low temperatures. Before hibernation, animals increase the proportion of unsaturated fatty acids in their cell membranes. This keeps the membranes flexible and functional, even when the body temperature drops dramatically. Without this change, the cell membranes will become stiff and lose their ability to function properly in the cold. The blood also changes before hibernation. Certain clotting factors are reduced, which helps prevent blood from clotting as it slows and cools during torpor. The kidneys adjust their function. The digestive system winds down. The intestines, which in an active animal are thick with absorptive cells and constantly processing food, become thin and simplified during hibernation. The body eliminates the metabolic cost of maintaining digestive machinery that will not be needed for months. Brown adipose tissue, commonly called brown fat, plays a crucial role in the hibernation cycle. Brown fat is a special type of fat tissue that is densely packed with mitochondria, the structures inside cells that generate energy. Unlike regular white fat, which stores energy for gradual release, brown fat is specialized for generating heat rapidly. When a hibernating animal begins an arousal, brown fat is one of the first tissues to become active. It burns at a high rate, generating the heat that warms the animal's body from the inside. In newborn human infants, brown fat also plays an important role in keeping the body warm, which is why hibernation research has some relevance to human medicine. The location where an animal hibernates is called a hibernaculum, from the Latin word for winter quarters. Different species use very different kinds of hibernacula. Ground squirrels dig elaborate burrow systems that may extend a meter or more below the surface. The depth provides insulation against the coldest temperatures above ground. The animal constructs a special hibernation chamber lined with dry grass and other insulating material. It seals the entrance to the burrow before entering torpor, creating a microenvironment that stays warmer than the surrounding soil. Woodchucks, also known as groundhogs or marmots depending on the species, dig similarly deep burrows and sleep alone through the winter. A woodchuck's burrow may have several chambers, including a separate toilet area that the animal uses during the brief arousals that punctuate its hibernation. The burrow is a remarkable piece of engineering, designed to maintain a stable temperature, exclude predators, and provide a safe space for the months-long pause in the animal's life. Bats hibernate in a variety of locations, including caves, mines, attics, and hollow trees. Caves are particularly important for many bat species because they maintain a stable temperature throughout the year, staying cool but not freezing even in the depth of winter. Bats that hibernate in caves often gather in large numbers, clustering together for warmth. A single cave may shelter hundreds of thousands of hibernating bats, their bodies pressed together, their metabolisms barely ticking over, their wings folded tightly around them. The little brown bat, one of the most common hibernating bats in North America, may hibernate for as long as 6 months. During hibernation, its body temperature drops from around 40 degrees Celsius to just a few degrees above the temperature of the cave it occupies. Its heart rate falls from around 1,000 beats per minute when active to as few as 20 during torpor. Its breathing slows to just a few breaths per minute. A bat in deep torpor can appear, to a casual observer, to be dead, but placed in a warm environment it will slowly begin to warm, and within an hour or two it will be flying. Bats face a particularly severe challenge from a disease called white nose syndrome, which is caused by a fungal pathogen called Pseudogymnoascus destructans. This fungus grows on the skin and wings of hibernating bats, causing irritation that triggers more frequent arousals. The extra arousals deplete the bat's fat reserves, often causing the animal to starve to death before spring. White nose syndrome has killed millions of bats in North America since it was first detected in a cave in New York in 2006. It represents one of the most severe wildlife disease events ever recorded in North America, and it has drawn significant scientific attention to the biology of bat hibernation. Bears present one of the most interesting and complex cases in the study of hibernation. For many years, scientists debated whether bears actually hibernate at all, because their body temperature does not fall as dramatically as it does in ground squirrels or bats. A hibernating black bear may maintain a body temperature of around 30 to 35 degrees Celsius, compared to its normal temperature of around 38 degrees. This is a modest reduction compared to the near-freezing temperatures reached by the most extreme hibernators. For this reason, some scientists historically preferred to describe bears as entering a state of winter lethargy, or denning, rather than true hibernation. More recent research has shifted this view. Studies measuring the metabolic rate of hibernating bears have found that their metabolism drops to around 25% of their summer rate. This is a very substantial reduction, even if not as extreme as in a ground squirrel. Bears also show many of the other hallmarks of hibernation, including reduced heart rate, reduced breathing, and a cessation of eating, drinking, urinating, and defecating for months at a time. The fact that a bear can go 5 to 7 months without urinating or defecating is physiologically remarkable. In most mammals, waste products would build up to toxic levels within days. Bears have evolved biochemical mechanisms to recycle waste products, converting nitrogenous wastes that would normally be excreted into useful proteins that help maintain muscle mass during the long fast. A hibernating bear in its den is also capable of giving birth. Female bears that enter their dens pregnant will give birth in January or February while still in their sleep-like state. The cubs are born tiny and helpless, weighing only around 300 to 500 grams at birth. They nurse while the mother remains in her torpid state, growing rapidly on her rich milk. The mother's body continues to produce milk even while she is fasting, drawing on her fat reserves to sustain both herself and her growing cubs. By the time the family emerges from the den in spring, the cubs are large enough to follow their mother and begin learning to find food. The European hedgehog is one of the most familiar hibernating animals in Europe, and is a good example of how hibernation works in a small mammal that faces different challenges than a large bear or a burrowing squirrel. Hedgehogs hibernate from around October or November until March or April, depending on temperatures and food availability. Before hibernating, they must build up sufficient fat reserves. A hedgehog that is underweight when cold weather arrives may not survive the winter because it will exhaust its fat reserves before temperatures warm enough for it to find food again. Hedgehogs hibernate in nests made of leaves, grass and other plant material, often tucked under a log, a pile of garden debris, or in a dense thicket. The nest provides insulation against the cold. During hibernation, a hedgehog's body temperature falls to within a few degrees of the ambient temperature, often dropping to around 5 degrees Celsius. Its heart rate falls from around 190 beats per minute to as few as 20. It breathes once every few minutes. Like other hibernators, it arouses periodically, and these arousals are energetically costly. One of the risks for hibernating hedgehogs in the modern world is the autumn bonfire. In Britain and Ireland, large bonfires are traditionally built in autumn from piles of garden waste and leaves. Hedgehogs seeking hibernation sites sometimes take up residence inside these piles before the bonfire is lit. Wildlife organisations regularly remind people to check their bonfires before lighting them. This small detail illustrates how ancient biological behaviours intersect with the modern human world in unexpected ways. The common dormouse is another small European mammal that hibernates, and it is notable for spending a remarkable proportion of its life in torpor. Dormice may hibernate for as long as 7 or 8 months of the year, spending more time in hibernation than any other European mammal. The word dormouse may in fact derive from the French word dormir, meaning to sleep. In the tale of Alice in Wonderland, the dormouse at the tea party is perpetually falling asleep, a reflection of this animal's well-known sleepiness. During hibernation, a dormouse's body temperature can fall to around 1 degree Celsius. Its heart rate slows to just a few beats per minute. It curls into a tight ball, tucking its head beneath its tail for additional insulation. In North America, the jumping mouse is another remarkable small hibernator. Some species of jumping mouse may hibernate for as long as 9 months, longer than almost any other North American mammal relative to the length of the year. They prepare by eating intensively in late summer, nearly doubling their body weight. They then construct deep underground nests and enter a state of deep torpor that lasts through the entire winter and into late spring. A jumping mouse in deep hibernation is cold and stiff to the touch, and shows almost no visible signs of life. Reptiles and amphibians also entered states of dormancy during cold periods, though their situation is somewhat different from the hibernation of warm-blooded mammals. Reptiles and amphibians are ectotherms, meaning they rely on external heat sources rather than internal metabolism to regulate their body temperature. When temperatures fall, their body temperature falls with the environment, and their bodily functions slow accordingly. This state of cold-induced dormancy in reptiles is sometimes called brumation to distinguish it from the mammalian hibernation. A box turtle entering brumation in autumn will bury itself in the soil or leaf litter, where it will remain immobile until spring. Its metabolism slows to an extraordinary degree. It doesn't need to arouse periodically the way mammalian hibernators do, because its metabolic demands are so low that it can sustain itself on stored energy for months. without the complications that drive mammalian arousals. Its heart may beat only a few times per hour. Its oxygen consumption drops to nearly zero. Because turtles can absorb small amounts of oxygen through their skin and the lining of their cloaca, some aquatic turtles can remain submerged beneath the ice of frozen ponds throughout the winter, obtaining just enough oxygen from the cold water to sustain their barely ticking metabolism. Wood frogs take this to an even more extreme conclusion. The wood frog, which lives across much of North America, including regions well north of the Arctic Circle, survives winter by freezing solid. As winter approaches and temperatures fall, ice begins to form in the spaces between the wood frog's cells. As the ice spreads, it draws water out of the cells, concentrating the fluids inside them. At the same time, the frog's liver converts stored glycogen into glucose, which floods the cells and acts as a kind of antifreeze, preventing the intracellular fluid from freezing even as the spaces around the cells fill with ice. The frog's heart stops, its breathing stops, it becomes rigid and cold, essentially a frozen object. Yet when spring arrives and temperatures rise, the ice melts, the heart restarts, and the frog hops away to breed in the nearest pond. The wood frog can survive the freezing of up to 65% of the water in its body. It has no heartbeat, no breathing, and no brain activity during the frozen state, yet it is not dead. The biochemical machinery that allows it to survive this state has attracted considerable scientific interest, particularly from researchers studying organ preservation for transplant medicine. Understanding how the wood frog protects its cells during freezing could potentially lead to better methods for preserving human organs for transplantation. Insects enter states of dormancy during winter through a process called diapause. Diapause is a hormonally regulated pause in development or activity that allows insects to survive unfavourable conditions. Different species enter diapause at different life stages. Some insects overwinter as eggs, others as larvae, others as pupae, and still others as adults. The monarch butterfly is an interesting case that combines migration with a form of adult dormancy. Monarchs that emerge in late summer enter a state of reproductive diapause, in which their reproductive development is paused and their lifespan is extended dramatically. These autumn monarchs migrate to their overwintering grounds in Mexico, where they cluster in dense masses on olla miel fir trees, remaining relatively inactive through the winter before the warming of spring triggers the resumption of their reproductive development and their migration northward. Many other insects survive winter as adults in a state of cold torpor. The mourning cloak butterfly in North America is among the first butterflies to appear in spring, because it overwinters as an adult rather than as an egg or a pupa. It seeks shelter in tree cavities or beneath bark and enters a torpid state as temperatures fall. Like the wood frog, it can tolerate partial freezing, surviving temperatures that would kill most insects. When warm days arrive in late winter or early spring, it emerges to take advantage of early sap flows from trees. Honeybee colonies deal with winter differently from solitary hibernating animals. Rather than having individual bees hibernate, the entire colony acts as a superorganism that generates its own heat. As temperatures fall, the bees cluster into a tight ball, the winter cluster, inside the hive. The bees on the outside of the cluster are relatively cold and relatively inactive, while those of the centre generate heat by vibrating their flight muscles without moving their wings. The temperature at the centre of the cluster is maintained at around 20 to 30 degrees Celsius, even when outside temperatures fall far below freezing. The bees on the periphery and centre rotate positions so that no individual bee remains in the cold for too long. The colony survives on honey stored through the summer, consuming it gradually through the winter to fuel the heat-generating activity of the cluster. The study of hibernation has revealed unexpected connections to human medicine. One of the most active areas of research involves the mechanisms by which hibernating animals protect their brains during the dramatic physiological changes of torpor. During the deep cooling and warming cycles of hibernation, the synapses in the brain, the connections between nerve cells, undergo dramatic changes. In the early stages of torpor, synaptic connections retract. Dendrites, the branching extensions of nerve cells that receive signals, shrink and simplify. This retraction reduces the brain's need for energy. During each arousal, these synapses are rapidly rebuilt. This cycle of retraction and regrowth happens repeatedly through the winter, without causing any apparent permanent damage to the brain. This process has fascinated neurologists because it resembles in some ways what happens in the brains of people with certain neurological conditions. In Alzheimer's disease, synaptic connections are progressively lost in ways that are not reversed. Understanding why hibernating animals can safely dismantle and rebuild their synapses repeatedly without permanent damage might offer insights into how to protect human brains from similar damage. Research in this area is still in early stages, but it represents one of the more intriguing potential applications of hibernation biology to human health. Another medical application concerned the preservation of organs for transplantation. When a donor organ is removed from a donor and transported to a recipient, it must be kept viable during the time between removal and transplantation. Currently, organs are cooled to slow their metabolism and preserve them, but they can only be kept viable for a limited time, typically a few hours for a heart and up to a day for a kidney. Understanding the mechanisms that allow hibernating animals to survive prolonged cold and reduced blood flow could help researchers develop better methods for preserving donor organs, potentially extending the window for transplantation and saving more lives. Stroke research is another area where hibernation biology is relevant. During a stroke, blood flow to part of the brain is interrupted, depriving neurons of oxygen and causing them to die. Hibernating animals routinely experience dramatic reductions in blood flow during torpor, yet their brains survive intact. The mechanisms they use to protect their neurons during these episodes of reduced blood flow are of great interest to stroke researchers. Several compounds that appear to play protective roles in hibernating animals are being studied for their potential to protect human brain cells during strokes. The possibility of inducing a hibernation-like state in humans has been explored, most prominently in the context of long-duration space travel. A journey to Mars, for example, would take approximately 7 to 9 months. If astronauts could be placed in a hibernation-like state for much of that journey, the mission would require far fewer supplies. The astronauts would experience less psychological stress from confinement, and the radiation exposure of individual crew members could be reduced. Research into induced torpor for space travel has been ongoing for several years, and while human hibernation remains a significant physiological challenge, the basic concept is not considered entirely impossible. The hormone melatonin continues to interest researchers studying hibernation. Melatonin is produced by the pineal gland in the brain in response to darkness. Its production rises in winter when nights are long and falls in summer when nights are short. In many hibernating animals, melatonin plays a key role in triggering the seasonal changes that prepare the body for hibernation. It affects the timing of hyperphagia, the changes in fat deposition and the suppression of reproductive activity that characterize pre-hibernation preparation. Melatonin supplements are widely used by humans as a mild sleep aid, and while the doses used for human sleep are far lower than those that would be relevant to hibernation, the underlying biology that connects melatonin to seasonal cycles and sleep is shared across many mammals. The torpor that characterizes daily and seasonal hibernation is related to another phenomenon seen across a much wider range of animals, including hummingbirds, bats, and many small rodents. This phenomenon is called daily torpor or nightly torpor, depending on the animal's activity pattern. In daily torpor, an animal's body temperature and metabolism drop significantly for a period of hours, typically during the part of the day when the animal would normally be inactive. A hummingbird, for example, burns enormous amounts of energy during the day as it hovers at flowers and generates body heat. At night, rather than continuing to burn fuel at this rate while inactive, it enters a state of torpor in which its body temperature drops dramatically and its metabolic rate plummets. By morning, it has warmed back up and is ready to resume activity. Daily torpor is in some ways a miniature version of seasonal hibernation, using the same basic physiological mechanisms on a compressed time scale. The use of torpor extends even further into the animal kingdom at mammals and birds. Some species of fish are known to enter states of dormancy. Lungfish, ancient fish that breathe air as well as using gills, can seek the drying up of their watery habitats by burrowing into the mud and secreting a mucus cocoon around themselves. In this state, called estivation, they can survive for years without water, with their metabolism reduced to a tiny fraction of its normal rate. Estivation functions as the summer equivalent of hibernation, a dormancy triggered by heat and dryness rather than cold. The physiological principles overlap considerably with winter hibernation, and researchers studying both phenomena have found useful parallels between them. Some species of snail also estivate. Land snails retreat into their shells during dry or cold periods and seal the opening with a dried mucus called an epiphragm. In this sealed state, the snail's metabolism slows greatly, and it can survive for months or even years without food or water. Snails collected in natural history museums and placed in storage have sometimes been found years later to still be alive. The social context of hibernation varies considerably among species. Most mammalian hibernators are solitary in their winter dens, but some species hibernate in groups, and the grouping appears to provide thermal benefits. Yellow-bellied marmots, which live in mountainous areas of western North America, hibernate in family groups in underground burrows. The shared body heat of the group helps reduce the metabolic cost of maintaining minimum body temperatures. The young marmots of the year, which have smaller fat reserves than adults, benefit particularly from the warmth of the group. Isolated young marmots have lower survival rates during winter than those that hibernate in family groups. Prairie dogs, though they do not enter true hibernation, reduce their activity and spend most of the winter in their underground burrow systems. Their tunnels and chambers provide insulation, and their social groupings contribute to warmth. The distinction between prairie dogs, which remain somewhat active underground in winter, and true hibernators like ground squirrels, which enter deep torpor, illustrates the spectrum of winter strategies that exist even among closely related animals. The brown bear and the polar bear represent interesting points on the spectrum. The brown bear, as discussed earlier, enters a winter den and reduces its metabolism substantially, but does not enter the deeper states of torpor. The polar bear has a more variable relationship with winter denning. Female polar bears that are pregnant excavate snow dens and spend the winter in a state of reduced activity, giving birth to cubs in the den. Male polar bears and non-pregnant females remain active through the winter, continuing to hunt seals on the sea ice. This species-level variation in winter strategy reflects the different environmental pressures on different individuals within the same species. The timing of emergence from hibernation is as carefully regulated as the timing of entry. Emergence is triggered by rising temperatures, increasing day length, and the internal biological clock that has been ticking quietly throughout the winter. In obligate hibernators, the timing of emergence is under strong hormonal control. Testosterone levels in male ground squirrels begin to rise before they emerge from hibernation, and males typically emerge several weeks before females. This early emergence gives males time to establish territories and compete for mates before the females appear. The females, when they emerge, are immediately ready to mate, and the breeding season is brief and intense. But the condition of an animal upon emerging from hibernation depends largely on how much fat it had stored before entering and how many arousals it underwent during the winter. Animals that entered hibernation with insufficient fat reserves, or that experienced unusually cold winters requiring more metabolic effort to maintain a minimum body temperature, or that underwent more frequent arousals than usual, may emerge in a weakened state. These animals must find food quickly to survive. The first weeks after emergence are a critical period for many hibernators, particularly young animals, and those that entered hibernation underweight. Climate change is affecting hibernation patterns in ways that researchers are only beginning to document. Rising temperatures are causing many hibernating species to emerge from hibernation earlier in spring. This can create a mismatch between the timing of emergence and the availability of food. If a hibernating animal emerges before the plants and insects that it relies on are available, it may face a period of food scarcity at a time when its fat reserves are already depleted. Some studies have found that ground squirrel populations in areas with rapidly warming springs are experiencing higher mortality rates because the animals are emerging before sufficient food is available. At the other end, warmer autumns are shortening the period available for pre-hibernation hyperphagia. If the food that animals rely on to build their fat reserves is available for fewer weeks before cold weather sets in, some animals may enter hibernation underweight. Research on hibernating bears in the American Southwest has found evidence of reduced fat reserves in some populations, potentially related to changes in the availability of autumn foods driven by changing precipitation patterns. Some hibernating species are responding to warming temperatures by shifting their range to higher elevations or higher latitudes, following the cooler temperatures that their biology requires. Picus, small relatives of rabbits that live on rocky mountainsides in western North America and do not truly hibernate but cache food for winter, are among the animals most affected by warming mountain temperatures. As temperatures rise, the cool rocky habitats picas require are moving higher up the mountains. In some mountain ranges, picas have already lost much of their historical habitat and cannot move higher because they have reached the mountaintop. The study of hibernation also offers insights into the aging process. Hibernating animals show some intriguing patterns related to longevity. Small mammals generally age much faster than large mammals, and small hibernators age more slowly than would be expected for their size. A little brown bat, which weighs only a few grams, can live for more than 30 years, which is extraordinary for an animal of its size. Most non-hibernating small rodents live only a year or two. Researchers have proposed that the periods of reduced metabolic rate during hibernation may slow the accumulation of cellular damage that contributes to aging. During deep torpor, free radical production, which contributes to cellular aging, falls dramatically. The cold temperatures of torpor also slow oxidative damage to cellular components. If the time spent in torpor effectively pauses the aging process, then long hibernating species may accumulate fewer years of physiological wear than their chronological age would suggest. This hypothesis has been tested in several ways. Studies of bat longevity have found that bat species that spend more time in torpor tend to live longer, even after accounting for factors like body size and predation risk. Similar patterns have been noted in ground squirrels. The animals that hibernate for the longest periods tend to have the longest lifespans. While the relationship is not simple, and many other factors influence longevity, the correlation between hibernation duration and lifespan is consistent enough to suggest that torpor does indeed slow some aspects of biological aging. The mechanics of how hibernating animals prevent muscle loss are of interest to researchers studying human muscle wasting diseases. During extended periods of inactivity, most mammals, including humans, lose significant muscle mass. This process is called disuse atrophy. Patients confined to bed rest can lose a substantial proportion of their muscle mass in a matter of weeks. Hibernating bears can remain immobile for 5 to 7 months and emerge in spring with remarkably little muscle loss. Their nitrogen recycling mechanisms, which allow them to convert waste products into proteins, appear to play a key role in maintaining muscle mass. Understanding these mechanisms could potentially help develop treatments for muscle-wasting conditions in humans. The genome of hibernating animals has been studied extensively in the hope of identifying the specific genes that control the hibernation process. What researchers have found is that hibernation does not appear to require unique genes that are absent from non-hibernating mammals. Instead, the same genes found in non-hibernating mammals are expressed differently in hibernating ones. The regulation of gene expression, rather than the presence of unique genes, appears to be the key difference. This is, in a sense, reassuring for researchers interested in applying hibernation biology to human medicine. It suggests that the same basic genetic toolkit present in humans might, in principle, be manipulated to produce hibernation-like states, although the practical challenges of doing so are enormous. Epigenetics, the study of changes in gene expression that do not involve changes in the DNA sequence itself, has become an important framework for understanding hibernation. Hibernating animals show dramatic changes in epigenetic markers as they move between active and torpid states, ethyl groups attached to DNA and chemical modifications to the proteins that package DNA can turn genes on or off without changing the underlying genetic code. The seasonal cycle of hibernation involves waves of epigenetic change that alter which genes are active in different tissues at different times of year. Unravelling the details of these epigenetic programs is an active area of research that promises to illuminate both the biology of hibernation and the broader field of epigenetic regulation. The fat that hibernating animals accumulate before winter is not a single uniform substance. White adipose tissue, which is the common form of fat found in most mammals, stores energy in the form of large fat droplets. It has relatively few mitochondria and generates heat only slowly. Brown adipose tissue, as mentioned earlier, is specialized for rapid heat generation. Most mammals also have a form of fat called barge or brite fat, which can be induced to behave more like brown fat under certain conditions. The balance between these different types of fat tissue and the specific fatty acids stored within them varies seasonally in hibernating animals and is carefully tuned to support both the extended fasting of torpor and the rapid warming of arousal. The specific composition of the stored fat is also important. Polyunsaturated fatty acids, particularly those rich in omega-3 fatty acids, are preferentially stored before hibernation in many species. These fatty acids remain fluid at lower temperatures than saturated fats do, and their presence in stored fat may help maintain the fluidity of fat deposits even when body temperature falls dramatically. Some research suggests that the diet animals eat in the weeks before hibernation influences not just how much fat they store, but also the composition of that fat and, by extension, the quality of their hibernation. Animals that have access to foods rich in omega-3 fatty acids before hibernation may enter torpor more easily and arouse more successfully than those that do not. The kidneys of hibernating animals undergo remarkable changes that allow them to survive months without urination. In a normally active mammal, the kidneys continuously filter blood, removing waste products and excess water, which are excreted as urine. If this process was simply to stop during hibernation, waste products would build up in the blood and become toxic within days. Instead, hibernating animals downregulate kidney function in a carefully controlled way. The filtration rate of the kidneys drops enormously, but some minimal filtration continues. Waste products that do accumulate are processed in ways that differ from the normal excretory pathway. In bears, urea, the main nitrogenous waste product, is recycled by bacteria in the gut and converted into amino acids that can be used to build proteins. This recycling loop allows bears to maintain muscle and organ proteins during their long fast, while producing almost no urine. The role of the gut microbiome in hibernation is an emerging area of research. The trillions of bacteria that live in the digestive tract of mammals play important roles in digestion, immune function, and metabolism. During hibernation, the composition of the gut microbiome changes dramatically. In some species, the microbiome appears to shift toward populations of bacteria that are more effective at recycling nutrients and producing compounds that support the host's survival during fasting. Research in ground squirrels has shown that the microbiome undergoes seasonal cycles that parallel the hibernation cycle, with different bacterial populations dominating in summer and winter. Transferring the winter microbiome of a hibernating ground squirrel into a non-hibernating relative produced some metabolic changes similar to those seen in hibernation, suggesting that the gut bacteria themselves contribute to the metabolic program of hibernation, rather than simply responding passively to changes in their host. The pineal gland, which produces melatonin in response to darkness, is not the only part of the brain involved in regulating hibernation. The hypothalamus, a small but critical region at the base of the brain, plays a central coordinating role. The hypothalamus receives information about day length, temperature, and the body's energy stores, and it integrates these signals to regulate the timing and depth of hibernation. It communicates with the rest of the body through both hormonal signals and direct neural connections. Lesions in certain parts of the hypothalamus in ground squirrels can disrupt the normal hibernation cycle, either preventing animals from entering torpor or causing them to fail to arouse properly. Adenosine, a molecule that accumulates in the brain during wakefulness and drives the feeling of sleepiness, also plays a role in the initiation of torpor. Adenosine signaling the brain increases as an animal prepares for hibernation, contributing to the shift toward inactivity and the deepening of torpor. Drugs that block adenosine receptors in the brain, such as caffeine, which works partly by blocking adenosine receptors in humans, can in some cases delay or reduce torpor in hibernating animals, providing an interesting if indirect parallel between caffeine's effects on human sleep and its effects on animal hibernation. The cardiovascular system of hibernating animals shows adaptations that allow it to function at extremely low temperatures and reduced rates without sustaining damage. The heart muscle of a hibernating ground squirrel continues to beat even when the body temperature is near zero, a feat that is not possible for the heart muscle of most non-hibernating mammals, which would stop beating and suffer damage at such temperatures. Studies of the proteins that regulate heart muscle contraction in hibernating animals have found that these proteins are modified in ways that allow them to function efficiently across a very wide temperature range. Understanding these modifications is of interest to cardiac researchers who are seeking ways to preserve the heart during cardiac surgery, which sometimes requires cooling the heart to very low temperatures. The bone density of hibernating animals is another area that has attracted medical interest. In humans and most mammals, extended immobility causes a loss of bone density, as the mechanical forces that normally stimulate bone maintenance are absent. This is a serious problem for astronauts in zero gravity, and for patients confined to prolonged bed rest. Hibernating bears, despite months of complete immobility, show remarkably little loss of bone density over the winter. They emerge from their dens in spring with bones that are essentially as strong as when they went in. The biochemical mechanisms that allow bears to maintain their bones during hibernation are not fully understood, but they appear to involve the continued activity of bone-building cells, osteoblasts, at a level that compensates for the reduced mechanical stimulation. Identifying the signals that maintain osteoblast activity in hibernating bears could potentially lead to treatments for osteoporosis and other bone loss conditions in humans. The lungs of hibernating animals face the challenge of functioning efficiently across a very wide range of breathing rates. During active periods, a ground squirrel breathes many times per minute. During deep torpor, it may breathe only once every few minutes. The lungs must be able to sustain gas exchange at both extremes. Researchers found that the surface tension properties of the fluid lining the alveoli, the tiny air sacs in the lungs where gas exchange occurs, change during hibernation in ways that prevent the alveoli from collapsing when breathing is very slow and shallow. These changes involve modifications to the surfactant proteins that coat the alveolar surface, and they represent another example of how hibernation requires precise adjustments across multiple organ systems working in coordination. The liver plays a central role in the metabolic transformations of hibernation. It is the liver that converts stored glycogen into glucose for the wood frog's anti-freeze protection. It is the liver that processes fatty acids released from adipose tissue and converts them into ketone bodies, which serve as a fuel source for the brain and other organs during the fat-fueled fast of hibernation. It is the liver that recycles nitrogenous wastes in bears. During the transition from active to torpid states, the liver undergoes dramatic changes in gene expression, shifting from its summer program of processing dietary nutrients to its winter program of metabolizing stored fat and recycling waste products. These liver programs are among the best studied aspects of hibernation biochemistry. The retinas of hibernating animals experience changes during torpor that would normally be considered signs of damage. In most mammals, extreme cold causes the retinal cells to degenerate. Yet hibernating ground squirrels can allow their retinal temperature to fall dramatically during torpor without suffering permanent visual damage. the retinas recover fully during each arousal. The mechanisms that protect the retina during these cold cycles are of interest to ophthalmologists studying conditions like age-related macular degeneration and other retinal diseases in which cellular damage leads to vision loss. The remarkable resilience of hibernating animals in the face of extreme physiological challenges has led some researchers to describe hibernation as a kind of natural stress test. The animals that survive multiple hibernation cycles are demonstrating the robustness of their biology. Each cycle of cooling and warming, each extended fast, each period of near zero metabolism is a challenge that the body meets with a suite of carefully evolved adaptations. The fact that hibernating animals can undergo those challenges repeatedly, year after year, without apparent cumulative damage speaks to the elegance of this biological machinery involved. As the science of hibernation continues to advance, researchers are increasingly able to identify the molecular signals that initiate, sustain, and end the hibernation state. One candidate molecule that has received considerable attention is a compound called delta sleep-inducing peptide, or DSIP. This small protein, first identified in the blood of sleeping rabbits, has been found at elevated levels in the blood of hibernating animals during torpor. When injected into non-hibernating mammals, it can induce states that resemble torpor. However, DSIP is almost certainly only one component of a complex signaling system, and its precise role in natural hibernation remains an area of active investigation. Another molecule of interest is hydrogen sulfide, a gas that is toxic at high concentrations, but that plays signaling roles at very low concentrations in many biological systems. Researchers have found that very low doses of hydrogen sulfide can induce a suspended, animation-like state in mice, reducing their metabolic rate and lowering their body temperature. The effect is reversible, and mice recover fully when the hydrogen sulfide is removed. This finding, published in 2005, generated considerable excitement and prompted speculation about whether hydrogen sulfide could be used to induce a human hibernation-like state for medical purposes. Subsequent research has shown that the effects of hydrogen sulfide are more complex and species-specific than initially hoped. But the underlying biology remains of interest. The relationship between hibernation and immune function is another area of active research. As noted in the discussion of bats, the immune system is substantially suppressed during deep torpor. This suppression is not simply a passive consequence of reduced metabolism, it appears to be an active part of the hibernation program. The inflammatory responses that form a key part of the immune system's defense against pathogens require energy and produce cellular damage as side effects. During hibernation, when energy is scarce and priority is conservation, suppressing inflammation may be beneficial even at the cost of reduced resistance to infection. The periodic arousals that hibernating mammals undergo may serve partly to allow brief windows of immune activity, as the immune system takes advantage of the warming period to conduct maintenance and surveillance. The discovery that some hibernating animals can survive freezing, and the related finding that others can tolerate near-freezing temperatures without lasting damage, has stimulated research into cryoprotection across many fields. Cryoprotection refers to the ability of tissues and cells to survive very low temperatures without damage. The wood frog's use of glucose as an intracellular cryoprotectant is one example. Other animals use different cryoprotective strategies, including the production of specialized antifreeze proteins that bind to ice crystals and prevent them from growing to damaging sizes. These antifreeze proteins were first discovered in Antarctic fish, which must survive in waters that are below the normal freezing point of fish blood. Similar proteins have since been found in a variety of cold-adapted organisms, including insects, plants, and some microorganisms. The study of hibernation also intersects with research on the nature of consciousness. When a mammal is in deep torpor, is it conscious in any meaningful sense? Brain activity is minimal. The animal does not respond to sensory stimuli, yet it is not dead, and the brain is not permanently altered. When the animal arouses, it returns to a fully functional state. The question of what, if anything, is experienced during deep torpor is one that current science cannot answer directly. The subjective experience of an animal in torpor, if any experience exists at all, remains beyond the reach of current measurement techniques. What can be said is that the brain in deep torpor is in a profoundly different state from the brain in normal sleep, and profoundly different from the brain in wakefulness. Whatever experience, if any, accompanies that state, it represents a biological condition that has no direct parallel in the lives of non-hibernating mammals. Ground squirrels have served as important laboratory models for hibernation research, partly because of the practical ease of working with them, and partly because of the extreme nature of their hibernation. But they are not the only mammals that have been brought into laboratory settings for hibernation research. Syrian hamsters, also called golden hamsters, are a domesticated species that retains its ancestral ability to hibernate under the right conditions. Wild Syrian hamsters hibernate in their native Middle Eastern habitats, but most individuals raised as pets are kept warm enough that they never enter torpor. When placed in cold temperatures with reduced food and short day lengths, captive Syrian hamsters will enter torpor, making them a useful and accessible laboratory model for hibernation research. The 13-lined ground squirrel, native to the grasslands of central North America, has become one of the most important laboratory models for hibernation genetics and physiology. Its genome has been sequenced, and researchers have used it to map the changes in gene expression across the full annual cycle of hibernation, from the active summer phase through hyperphagia, entry into torpor, deep hibernation, arousal, and emergence. These studies have generated vast datasets that continue to be analysed for insights into the molecular biology of hibernation. The role of temperature in triggering hibernation is sometimes misunderstood. Temperature is an important environmental cue for many hibernating species, but it is not the sole trigger. Day length, food availability, and internal biological clocks all contribute to the timing of hibernation. In some species, the internal clock is so powerful that animals will attempt to hibernate even under laboratory conditions with constant temperature and artificial light, though the rhythm may drift somewhat without the normal environmental cues. The integration of internal and external signals to produce the precise seasonal timing of hibernation is one of the more sophisticated aspects of the biology of these animals. The nests and dens of hibernating animals are not passive shelters. They are functional microenvironments that the animals construct with care to optimize the conditions for their long sleep. Nest insulation affects the rate at which body heat is lost to the environment, which in turn affects the metabolic cost of maintaining a minimum body temperature. A better insulated nest requires less metabolic effort to stay warm, which translates into more fat reserves remaining when spring arrives. Studies of nest construction behavior in small hibernators have shown that animals carefully select nesting materials for their insulating properties. Dry leaves and grasses are preferred over wet or decomposing materials. The depth of the burrow is also carefully calibrated, deep enough to stay below the frost line, but not so deep that building and maintaining it requires excessive effort. The behaviour of animals in the weeks leading up to hibernation reflects the urgency of preparation. Hyperphagia dominates, but there are also behaviours related to site selection, nest construction, and social interactions. In territorial species, the boundaries of individual territories may shift in autumn, as animals move to access the food sources they need for hyperphagia. Conflicts over food resources can be more intense than at other times of year. The outcome of these conflicts, which animals successfully build sufficient fat reserves, and which do not, can determine who survives the winter and who does not. In colonial bat species, the movement from summer roosting sites to hibernacula is a significant seasonal event. Bats may travel considerable distances from their summer territories to their hibernation caves, guided by a combination of listened knowledge and navigational abilities that are not yet fully understood. The same caves are used year after year by many bat species, with some hibernacula known to have been in continuous use for centuries. The fidelity of bats to their hibernation sites reflects the scarcity of suitable caves and the value of a known reliable winter refuge. The sounds of a hibernation cave in winter are very different from those of the same cave in summer. In summer, a bat cave is a place of intense activity, filled with the high-frequency calls of thousands of individuals navigating in the dark, the sounds of pups calling to their mothers, and the general noise of a dense animal colony. In winter, the same cave is nearly silent. The hibernating bats hang motionless from the ceiling, their bodies slightly fuzzy with condensation from the cool air. The only sounds are the slow, irregular drips of water and occasionally the faint rustle of a bat shifting position slightly during an arousal. The beauty of hibernation as a biological strategy lies in its elegance. It is, at its core, a solution to a problem that every living thing faces: the need to match energy expenditure to energy availability. When energy is scarce and energy expenditure cannot be reduced through behavioural means alone, some animals have evolved the ability to reduce their energy expenditure at a physiological level to slow their bodies to a bare minimum and wait for better conditions. The strategies they use are diverse, the timing is precise, the mechanisms are complex, and the results, whether in the frozen wood frog, the barely breathing bear, or the cold and still bat, are a testament to the capacity of evolution to produce solutions of extraordinary sophistication to the challenges of survival. As you rest and allow these ideas to settle, consider the remarkable fact that across the world, in burrows and caves and hollow trees and frozen ponds, countless living things are right now in the midst of their long winter pause. Their hearts are barely beating. Their breath is barely moving. Their temperatures have fallen to the edge of what life can sustain. And yet they are not gone. They are waiting as they have waited every winter, through tens of millions of years of winter, for the warmth and light that will, in time, return. It is a quiet kind of persistence, a patient kind of survival, and it is happening right now in the dark and cold all around the sleeping world. The hibernation of small rodents has been studied in connection with the science of circannual rhythms, which are biological cycles that repeat approximately once per year. These rhythms are generated internally by biological clocks that run independently of environmental cues Though they're normally synchronized to the actual year by light and temperature signals, circannual rhythms have been demonstrated in a number of hibernating species by keeping animals in constant laboratory conditions for multiple years and observing that they continue to cycle between active and torpid states at roughly annual intervals. The internal clock drifts slightly, without environmental cues, completing its cycle in slightly more or less than 365 days, but the rhythm persists. This demonstrates that the timing of hibernation is not simply a passive response to environmental conditions. It is driven by an internal program that measures time independently of the outside world. The mechanisms underlying circannual clocks are not yet fully understood, but research suggests that they may involve some of the same molecular components as the circadian clock, the daily biological clock that governs sleep and wakefulness in most animals. The circadian clock is generated by a feedback loop of genes and proteins that cycle over approximately 24 hours. The circannual clock may involve a similar feedback loop operating on a much longer timescale, though the precise molecular details remain an active area of investigation. The way in which hibernating animals assess their own body fat stores and make decisions about when to begin and end hibernation is another fascinating aspect of the biology. The hormone leptin, which is produced by fat cells and signals the brain about the body's energy stores, plays a role in regulating the timing of hyperphagia and hibernation entry in several species. When fat stores are high, leptin levels are elevated, and this signal helps suppress hunger in non-hibernating mammals. In hibernating mammals, however, the relationship between leptin and appetite is reversed during hyperphagia. Animals preparing for hibernation continue to eat voraciously, despite high leptin levels, suggesting that their brains have become temporarily resistant to leptin's hunger-suppressing effects. This transient leptin resistance is thought to be hormonally regulated, allowing the animal to override its normal satiety signals and continue eating until it has accumulated sufficient fat reserves. Once the animal has accumulated enough fat and entered hibernation, leptin levels fall as fat is consumed during torpor. The falling leptin signal is thought to be one of the factors that eventually contributes to arousal, as the brain detects that fat reserves are running low. The interplay between leptin, fat stores, and the timing of hibernation events illustrates the precision with which the body monitors and responds to its own energy state. The thyroid gland, which produces hormones that regulate metabolic rate throughout the body, undergoes changes during hibernation that contribute to the overall reduction in metabolism. Thyroid hormone levels fall during torpor, reducing the metabolic rate of virtually every tissue in the body. This is distinct from and complementary to the direct effects of cold temperature on chemical reaction rates. Both the hormonal reduction in metabolism and the temperature-driven slowing of chemical reactions contribute to the overall metabolic suppression of hibernation. When the animal arouses, thyroid hormone levels recover, helping to restore normal metabolic rates as body temperature rises. The adrenal glands, which produce stress hormones, including cortisol and adrenaline, also cycle seasonally in hibernating animals. In autumn, cortisol levels rise, contributing to the physiological changes that prepare the body for hibernation, including the mobilization of energy stores and the adjustment of immune function. During deep torpor, cortisol levels fall to very low levels. The absence of elevated cortisol during hibernation is in some ways counterintuitive, because the extreme physiological conditions of torpor might seem like a stress that would trigger a cortisol response. Instead, the hibernating body appears to have shifted into a mode in which the normal stress response is deliberately suppressed, allowing the body to maintain the deep torpor state without the metabolic costs of a stress response. The insulin system also undergoes changes during hibernation. Insulin, produced by the pancreas, normally regulates the uptake of glucose by cells and the storage of nutrients after meals. During hibernation, when the animal is fasting and relying on fat rather than glucose as its primary fuel, insulin sensitivity and secretion are altered. Some hibernating species appear to develop a state of insulin resistance during torpor, in which cells become less responsive to insulin. This resistance may help direct glucose away from peripheral tissues and reserve it for the brain, which depends more heavily on glucose than most other organs. The regulation of glucose metabolism during hibernation is relevant to research on type 2 diabetes, a condition in which insulin resistance plays a central role, and hibernating animals have been studied as models for understanding how the body manages insulin resistance without the harmful consequences seen in diabetes. The kidneys undergo one of the most dramatic functional changes during hibernation of any organ. The glomerular filtration rate, which measures how quickly the kidneys filter blood, falls to a tiny fraction of its normal value during deep torpor. Yet the kidneys do not fail. The reduced filtration is precisely controlled and coordinated with the overall reduction in metabolic activity. The tubules of the kidney, which normally perform the complex work of selectively reabsorbing useful substances and excreting waste, also adjust their function. Some studies have found evidence of structural changes in kidney cells during hibernation, including changes in the arrangement of cellular proteins involved in transport. These structural changes are reversed during arousal, and the kidneys return to full function within hours of the beginning of warming. The spleen, which serves as a reservoir of red blood cells and plays roles in immune function, also shows seasonal changes in hibernating animals. In some species, the spleen contracts during torpor, releasing stored red blood cells into the circulation. This may help compensate for the reduced heart rate and cardiac output during torpor, ensuring that sufficient oxygen carrying capacity is maintained even when blood flow is greatly reduced. The study of hibernation in bears has been advanced enormously by the development of remote monitoring technology. Early studies of bear hibernation required researchers to enter dens, often at considerable personal risk, to collect physiological data from sedated animals. Modern studies use implantable biologgers, small electronic devices that can be surgically placed inside animals and that record heart rate, body temperature, activity, and other variables continuously throughout the hibernation season. These devices transmit data remotely, allowing researchers to follow the entire hibernation cycle without disturbing the animals. The detailed physiological records obtained from these devices have revealed the fine structure of bear hibernation in ways that were not previously possible, including the precise timing of heart rate fluctuations, the gradual cooling and warming of different body regions, and the subtle variations in metabolic rate that occur within a single day. The bears that have been most intensively studied for hibernation biology include the American black bear, the brown bear, and to a lesser extent the Asiatic black bear and the sun bear. The polar bear, as noted earlier, has a somewhat different hibernation pattern than its terrestrial relatives. The sun bear of Southeast Asia, which lives in a tropical environment, does not hibernate at all, because the constant warmth and food availability of the tropical forests makes hibernation unnecessary. This absence of hibernation in the sun bear combined with the presence of hibernation in its cold-adapted relatives, illustrates how hibernation is a facultative adaptation that has been lost or retained depending on the ecological pressures each species faces. The evolution of hibernation is a subject of considerable interest. Hibernation has evolved independently in multiple lineages of mammals, which suggests that the physiological toolkit required for hibernation is relatively accessible to mammalian evolution, rather than being an exotic capability that requires entirely novel biological machinery. Hibernation appears to build on mechanisms that are present in some form in most mammals. The capacity for daily torpor, which is widespread among small mammals, may have been an evolutionary precursor to seasonal hibernation. Species that already had the ability to enter short periods of daily torpor may have been better positioned to evolve the extended seasonal hibernation that characterizes true hibernators. Molecular phylogenetic studies have attempted to reconstruct the evolutionary history of hibernation across mammals. These studies suggest that hibernation has evolved and been lost multiple times in different mammalian lineages. Some lineages appear to have lost the ability to hibernate as they colonized environments where hibernation was not necessary, while others may have secondarily evolved hibernation after a period without it. The genetic basis of hibernation, which appears to involve the differential regulation of widespread mammalian genes rather than entirely novel genes, is consistent with the relatively frequent evolutionary origin and loss of hibernation across this mammalian family tree. The evolution of hibernation in birds is a separate and interesting story. Most birds do not hibernate. They cope with winter through migration, behavioral thermoregulation, or physiological adaptations that allow them to survive cold nights. However, one bird, the common porwheel of western North America, is known to enter a state of prolonged torpor that meets most of the criteria for a true hibernation. The porwheel was known to the Hopi people of the American Southwest, who called it Holchko, meaning the sleeping one, a recognition of its winter disappearance. Scientific confirmation of porwheel hibernation came in 1946, when a researcher found a torpid individual in a rocky crevice in the California desert and measured its dramatically reduced body temperature and absence of obvious breathing. The porwheel's hibernation is not as prolonged as that of ground squirrels or bats. It typically lasts a few weeks to a few months, and the body temperature does not fall as low as in the most extreme mammalian hibernators. But it is a genuine suspension of normal physiological activity driven by cold and food scarcity, and it represents the only clear case of avian true hibernation currently known to science. Whether other birds are capable of similar states under extreme conditions remains an open question. Many birds, however, engage in daily torpor. Hummingbirds, as mentioned earlier, are among the most dramatic examples. A ruby-throated hummingbird, weighing only around 3 grams, must consume roughly its body weight in nectar each day during the warm season to fuel its extraordinarily high metabolic rate. On cool nights or during periods of food scarcity, hummingbirds enter a state of nocturnal torpor, in which their body temperature drops from around 42 degrees Celsius to temperatures as low as 10 degrees or even lower. Their heart rate falls from around 1,200 beats per minute to fewer than 50. They hang motionless and appear, to an observer, to be dead. In the morning, they warm back up and resume their frantic activity. The hummingbird's daily torpor is not hibernation, but it uses the same basic physiological mechanisms on a compressed timescale. Swifts, small aerial birds that spend almost their entire lives in flight, enter torpor during cold spells when flying insects are scarce. Alpine swifts, which have been tracked via electronic tags attached to their bodies, have been found to remain continuously airborne for 250 days at a stretch during their African wintering period, feeding on aerial insects the entire time. During cold or stormy periods when insects are unavailable even in Africa, swifts enter brief torpor states to conserve energy. The versatility of the torpor response, which can serve both as a brief, overnight energy conservation strategy and as a months-long survival mechanism depending on the species and its circumstances, reflects the fundamental biological utility of reducing metabolic rate in times of energy scarcity. Marsupials, the pouched mammals of Australia and the Americas, have their own representatives among the hibernators. The fat-tailed dunart, a small carnivorous marsupial of Australian grasslands, enters daily torpor and can survive extended periods in this state when food is scarce. The eastern pygmy possum of Australia enters torpor during cold periods and can remain in a torpid state for weeks at a time, surviving on stored fat. These marsupial hibernators provide interesting comparative data for researchers studying the evolution of hibernation, because marsupials diverge from placental mammals very early in mammalian evolution. The presence of similar hibernation mechanisms in both marsupials and placentals suggests that the basic physiological toolkit for torpor was present in early mammals and has been conserved across a very long evolutionary history. Monotremes, the egg-laying mammals of Australia and New Guinea, including the platypus and the echidnas, also show torpor behaviour. The short-beaked echidna enters torpor during cold periods in southern Australia, with body temperatures falling to near ambient levels. The platypus has been reported to enter torpor under some conditions as well. The presence of torpor across all three major groups of living mammals, the monotremes, the marsupials and the placentals, strongly supports the hypothesis that some capacity for torpor was present in the common ancestor of all living mammals, and that this capacity has been elaborated into seasonal hibernation in some lineages while being lost, or retained in a simpler form in others. The discovery of hibernation-like states in some large dinosaurs has been proposed by a small number of researchers based on bone growth patterns and geographic distribution. Some non-avian dinosaurs lived at high latitudes in environments that would have experienced prolonged winter darkness and cold. The hypothesis that some of these animals may have entered seasonal torpor to survive the winter has been discussed, though the direct evidence is limited. Most paleontologists are cautious about this idea because the evidence from bone growth patterns is ambiguous and can be interpreted in multiple ways. Nevertheless, the question of how large-bodied animals managed polar winters hundreds of millions of years ago remains an interesting one, and hibernation is at least plausible as one possible strategy. The connection between hibernation and the broader science of biological timekeeping is a rich one. Hibernating animals must maintain accurate knowledge of the time of year, even while their internal physiology is dramatically altered. The circadian clock continues to run during hibernation, ticking away even while the animal is in deep torpor. Somehow, the biological machinery that measures the passage of the year continues to function, even when the rest of the body is barely operating. How this clock maintains its accuracy during hibernation, when normal metabolic activity is suppressed and normal temperature cycles are absent, is not fully understood. One possibility is that the clock runs more slowly during torpor and is reset during each arousal, with the cumulative time in arousal serving as the measure of elapsed time. Another possibility is that the clock runs continuously at a reduced rate during torpor and uses compensatory mechanisms to maintain accuracy despite the temperature-induced slowing of chemical reactions. The process of emergence from hibernation is not simply a reversal of the process of entering it. While there are parallels, the biochemical and physiological changes that occur during emergence have their own specific character. As the animal begins to warm, brown adipose tissue becomes active and generates heat. Blood flow to the extremities gradually resumes. The heart rate climbs in stages. Breathing becomes more rapid and regular. Brain activity increases. The animal begins to shiver, which generates additional heat through muscle activity. The eyes open, the senses return. For a small hibernator like a ground squirrel, the entire process of warming from near freezing temperatures to normal body temperature takes approximately 2 to 3 hours. For a large hibernator like a bear, the process of fully emerging from winter torpor may take days or weeks, as the animal gradually increases its activity level. The first acts of a newly emerged hibernator often relate directly to the most pressing physiological needs. Animals that have not urinated for months or have produced very little urine may urinate soon after emerging. Animals that have not eaten for months begin foraging immediately. The type of food sought immediately after emergence is often dictated by what is available and by the specific nutritional deficits that have accumulated during the fast. Bears emerging in spring often seek out early sprouting vegetation, partly for its high water content and partly for nutrients that help the digestive system return to function after months of disuse. The period immediately following emergence is associated with some increased risk of predation for small hibernators. After months of inactivity, muscles may be slightly weaker and reflexes somewhat slower than in fully conditioned active animals. The animals are also focused intently on finding food, which can make them less vigilant against predators. Some research has found slightly elevated predation rates on ground squirrels and similar animals in the first days after emergence from hibernation, compared to later in the active season when they are fully conditioned. The reproductive urgency that drives male ground squirrels to emerge weeks before females reflects a broader pattern of sexual selection and reproductive strategy. Males that emerge early have more time to establish territories and accumulate the resources needed to compete for mates. But early emergence also carries risks. The food available in early spring may be limited, and the risk of another cold snap is real. Male ground squirrels that emerge too early may face cold conditions and food scarcity that drain their fat reserves before females emerge. The timing of male emergence appears to represent an evolved balance between the reproductive benefits of early emergence and the survival risks it entails. Female ground squirrels and other hibernators with delayed female emergence may benefit from the protection that male competition provides. By the time females emerge, males have already sorted themselves into a dominance hierarchy, and the dominant males are positioned to fertilize the most females in the brief breeding season. Females can assess male quality based on the outcomes of male-male competitions, potentially choosing mates with superior traits without having to engage in extended courtship. This compressed competitive breeding system is a consequence of the shortened active season that hibernation imposes. The young of hibernating species face particular challenges. Born in spring or early summer, they have only a few months in which to grow, learn to find food, and accumulate the fat reserves needed for their first hibernation. Young animals entering their first hibernation are at greater risk of not surviving than adults entering subsequent hibernations, because they have had less time to accumulate experience finding food, and because they are completing their growth while simultaneously preparing for hibernation. In years when food is scarce in late summer and autumn, the first-year survival rate among young hibernators can be substantially lower than in years with abundant food. The science of hibernation intersects with conservation biology in several important ways. Many hibernating species are threatened by habitat loss, climate change, disease, and other human-caused pressures. Understanding the specific physiological requirements of hibernating species, including the specific conditions they need in their hibernacula, the food sources they depend on for hyperphagia, and the temperature regimes that allow them to complete successful hibernation cycles, is essential for designing effective conservation strategies. For bat species threatened by white nose syndrome, understanding the conditions that favour or inhibit the growth of the causative fungus and the physiological responses of bats to infection is central to developing management strategies. For many cave-dwelling bat species, the hibernacula themselves are the bottleneck resource that limits population size. If a species depends on a small number of suitable caves for hibernation, and those caves are disturbed by human visitation, mining, or development, the entire regional population may be at risk. Conservation programs for hibernating bats often focus on identifying and protecting key hibernation caves, managing human access during the hibernation season, and working to prevent the spread of white nose syndrome between cave systems. The interaction between urban development and hibernating animals presents challenges and opportunities. Hedgehogs in European urban areas often adapt to the human environment, hibernating under garden sheds, in compost heaps, and beneath piles of vegetation. Urban green spaces can provide important foraging habitat for hedgehogs during the pre-hibernation hyperphagia period. Gaps in garden fences allow hedgehogs to move between urban green spaces, expanding their foraging range. Many urban wildlife organisations promote hedgehog-friendly gardening practices, including leaving areas of natural vegetation, avoiding slug pellets that can reduce the invertebrate prey hedgehogs depend on, and providing supplemental food in autumn to help underweight animals build sufficient fat reserves for hibernation. The scientific literature on hibernation is rich and rapidly expanding. New sequencing technologies have allowed researchers to examine the changes in gene expression during hibernation at an unprecedented level of detail. New imaging technologies allow the visualization of blood flow and metabolic activity in hibernating animals in ways that were previously impossible. New tracking technologies allow the movements and physiological states of hibernating animals in the wild to be monitored continuously. The combination of these new tools with the long history of detailed physiological and behavioral research on hibernating animals is generating a synthesis that is revealing the full complexity of this ancient biological strategy. The curiosity that drives this research is, at its core, a curiosity about life itself. What is the minimum that a living thing can do and still be alive? How far can the body's processes be slowed before they stop entirely? How does the body know when to slow down and when to speed up? How does it maintain the ability to restart after being so near to stillness? These questions are not only scientifically interesting, they touch on fundamental aspects of what it means to be alive. And hibernation, in all its quiet complexity, offers some of the most revealing answers available anywhere in the natural world. As these ideas drift through your thoughts, allow the pace of your own breathing to slow. The science of hibernation is, in a gentle way, an invitation to appreciate the capacity for stillness. Stillness is not the absence of life. In the hibernating ground squirrel, in the frozen wood frog, in the barely breathing bat, stillness is life at its most patient, its most enduring, its most deeply at rest. And rest, as the science of sleep and torpor both remind us, is not emptiness. It is preparation. It is the quiet gathering of resources for the return of warmth and activity and the long, full days ahead. For now, in this moment, allow your own thoughts to slow and soften. Let the facts settle like snow, and rest. The microhabitat within a hibernaculum can vary considerably across small distances, and hibernating animals often show highly specific preferences for particular locations within a given site. In a cave system used by hibernating bats, the distribution of individuals across different parts of the cave reflects subtle differences in temperature, humidity, air movement, and proximity to other bats. Some species prefer the warmest parts of the cave, where the metabolic cost of maintaining minimum body temperature is lowest. Others prefer cooler spots, where the deepest torpor possible at lower temperatures provides greater energy savings. Some prefer isolated crevices that offer protection from predators or from the disturbance of other bats. The patterns of distribution within hibernation sites are not random. They reflect individual choices made in response to the microenvironmental conditions that best serve each animal's physiological needs. The importance of humidity in bat hibernacula has been documented in connection with white nose syndrome. The fungus that causes white nose syndrome grows best in cool, humid conditions, which are precisely the conditions that many hibernating bats prefer. Bats in the coldest, most humid parts of caves are often most severely affected by the disease. Research has examined whether modifying the humidity of hibernacula, for example by improving ventilation to reduce humidity levels, could slow the spread of the fungus and reduce disease severity. The results have been mixed, because changes in cave conditions that reduce fungal growth may also alter the energy budget of hibernating bats in ways that are not always beneficial. The interaction between hibernating animals and their parasites presents another dimension of the hibernation biology. Many hibernating mammals carry populations of external parasites, including ticks, mites, and fleas. During hibernation, when the animal is cold and inactive, these parasites face their own survival challenges. Some parasites enter their own state of dormancy on the host animal during winter. Others abandon the host before hibernation and seek shelter in the nest material or surrounding environment. The nest and burrow of a hibernating animal can harbour a complex community of invertebrates, including both parasites and the beneficial organisms that help break down waste materials in the nest. Internal parasites also show seasonal patterns related to the host's hibernation cycle. Some intestinal parasites of ground squirrels show reduced population sizes during hibernation, as the slowing of gut function during torpor reduces the resources available to them. Others appear to time their life cycles to exploit the arousal periods when gut function is briefly restored. The relationship between host hibernation and parasite biology illustrates how hibernation creates ecological ripple effects that extend well beyond the hibernating animal itself. The sound environment of a hibernating animal's den is one of the few sensory channels through which information from the outside world can reach the dormant creature. While hibernating animals do not respond to most sounds during deep torpor, very intense vibrations can sometimes trigger arousal. This sensitivity is not a nuisance. It may serve as a last resort alarm system, allowing the animal to rouse and escape if a predator is actively excavating its burrow. The threshold for arousal in response to vibration decreases as hibernation depth increases, reflecting the balance between energy conservation, which requires resistance to unnecessary arousal, and survival, which requires some capacity to respond to extreme threats. The olfactory environment of the hibernation site may also carry information relevant to the hibernating animal. The den of a hibernating bear, for example, is known to retain the bear's own scent, which may play a role in site fidelity and in the recognition of the den in subsequent years. The scent of a predator near the den entrance might, in theory, be detected during an arousal and influence whether the bear remains in the den or emerges. The extent to which hibernating animals process olfactory information during the minimal arousal states is not well studied, but it represents an interesting question about the degree to which even hibernating animals maintain some low-level environmental awareness. The contribution of hibernating animals to ecosystem function is often underappreciated. Ground-burrowing hibernators, including ground squirrels and marmots, are significant ecosystem engineers. Their burrow systems aerate the soil, increase water infiltration, and redistribute nutrients. Their burrows also provide shelter for a wide variety of other animals, including birds that nest in abandoned ground squirrel burrows, snakes that overwinter in burrow systems, and insects that use the tunnels as refugia. The digging activity of these animals affects soil structure for large areas and contributes to the productivity of the grassland ecosystems they inhabit. The catching behaviour of some animals that do not fully hibernate also merits consideration alongside the hibernation story. Squirrels that do not hibernate, such as the grey squirrel and the fox squirrel of North America, survive winter by caching food in autumn and retrieving it during winter. These animals remain active through the winter, relying on memory and scent to locate their cached food. The contrast between the food caching strategy and the fat-storing hibernation strategy represents two distinct evolutionary solutions to the same problem of winter food scarcity. Both strategies have been successful, as evidenced by the widespread distribution and abundance of both hibernating and non-hibernating squirrel species. The pikas mentioned earlier in the context of climate change take a related but distinct approach. Pikas do not hibernate and do not store fat. Instead, they spend the summer and autumn cutting and drying vegetation, which they store in hay piles in their rocky dens. These hay piles serve as their food supply through the winter, and pika behaviour is focused on maximising the size and quality of the hay pile before winter arrives. A pika that fails to accumulate a sufficient hay pile will not survive the winter. The hay pile strategy required a different set of behavioural and cognitive adaptations than hibernation, but it achieves a similar end result: survival through a period of food scarcity without migration. The science of hibernation has benefited from the development of stable isotope analysis, a technique that allows researchers to trace the sources of materials in an organism's tissues. By analysing the ratios of different isotopes of carbon, nitrogen and other elements in the tissues of hibernating animals, researchers can determine what the animals have been eating, how much fat they have metabolized, and how their nitrogen balance has shifted over the course of hibernation. Stable isotope analysis has confirmed and quantified many of the physiological processes described in earlier research, adding a new level of precision to our understanding of the metabolic transformations that occur during hibernation. Telomeres, The protective caps at the ends of chromosomes that shorten with each cell division and are associated with aging have been studied in hibernating animals in the context of the hibernation longevity connection. Some research has found that hibernating animals show less telomere shortening over the course of a hibernation season than would be expected based on their chronological age, consistent with the hypothesis that torpor slows the aging process. However, the relationship between hibernation, telomere dynamics and longevity is complex, and not all studies have found consistent patterns. The question of how hibernation affects the molecular mechanisms of aging at the cellular level remains an active area of research. The study of protein aggregation during hibernation has revealed another dimension of the physiological challenges that hibernating animals face. In a number of neurodegenerative diseases, including Alzheimer's disease and Parkinson's disease, proteins that normally fold into specific three-dimensional shapes instead misfold and aggregate into clumps that disrupt cellular function. The cold temperatures of hibernation could, in principle, promote protein misfolding and aggregation. Yet hibernating animals emerge from torpor without signs of the protein aggregation that causes neurodegeneration in these diseases. Research into the mechanisms that protect proteins from misfolding during hibernation may therefore be relevant to understanding how to prevent or reverse protein aggregation in neurodegenerative diseases. The remarkable variety of animals that have evolved some form of hibernation or torpor reminds us that the pressure to solve the problem of winter food scarcity has been a powerful force in shaping the evolution of life. From the frozen wood frog to the barely breathing bat, from the denning bear to the dormant dormouse, from the estivating lungfish to the torpid hummingbird, the solutions that evolution has produced are diverse in their details, but unified in their fundamental logic. When conditions make survival through activity impossible, slow down, reduce the cost of being alive, wait, and when conditions improve, begin again. This logic is ancient. It predates mammals. It predates vertebrates. Single-celled organisms can enter states of dormancy that allow them to survive conditions that would kill active cells. Bacterial spores can persist for extraordinary lengths of time, resuming activity only when conditions become favourable. The dormancy of seeds allows plants to delay germination for years or decades until the right conditions arise. The desiccated, metabolically arrested form called a tun, adopted by tardigrades and certain nematodes under extreme conditions, is perhaps the most extreme example of a living thing, reducing its biological activity to the lowest possible level while retaining the ability to resume. Hibernation, seen in this context, is one expression of a principle that runs through all of life. The principle that survival sometimes requires waiting, and that waiting, done right, is itself a form of living. The quiet of winter, the stillness of the hibernating world, carries within it an enormous amount of biological activity, just slowed to a pace that is invisible to a casual observer. Beneath the frozen ground, hearts beat. Fat is consumed, molecule by molecule. Clocks tick. Cells maintain themselves at the edge of viability. The long pause is not empty. It is full of the slow, patient work of survival. And in spring, when the warmth returns and the ground softens and the first hungry animals emerge blinking into the light, all of that stored patience will suddenly transform into the urgent, vivid activity of the warm season. The contrast between the two is one of the most dramatic cycles in the natural world, and it is driven at its deepest level by the same biological logic that governs every living thing. Find energy, preserve energy, survive to use that energy when the time is right. Thank you for spending this time exploring the quiet science of animal hibernation. The world is full of sleeping things right now, each one a small marvel of biological engineering, each one waiting patiently for the return of warmth. May you find your own rest as deep and as restorative as theirs.